Yes, diet can profoundly rewire the gut's communication with the brain, and it does so by shaping the microbial ecosystem that interacts directly with gut sensors, known as neuropods. These specialized epithelial cells function like sensory neurons, translating information from nutrients and microbes into rapid electrical and chemical signals that reach the brain, often within milliseconds. What we eat determines which microbes thrive, which metabolites they produce, and how this symphony of signals is interpreted by the nervous system. This dynamic process influences not only digestion and hunger, but also emotional state, motivation, memory, and even our longer-term patterns of behavior around food. At the heart of this interaction is dietary fiber, a non-digestible carbohydrate that serves as the primary fuel for many beneficial gut bacteria. When fiber reaches the colon, it's fermented by microbes into short-chain fatty acids, SCFAs, such as butyrate, acetate, and propionate. These SCFAs interact directly with neuropods and enteroendocrine cells, stimulating the release of satiety hormones like GLP-1 and PYY, as well as neurotransmitters such as serotonin. The result is a cascade of signals that travel up the vagus nerve to appetite and emotion-related centers in the brain, helping to regulate meal size, hunger cues, and even feelings of well-being. But the effects of SCFAs go beyond fullness. They also strengthen the gut barrier, reduce inflammation, and modulate immune signaling, all of which improve the clarity and reliability of gut-brain communication. A well-fed microbiome rich in fiber-degrading species supports more consistent and adaptive signaling through neuropods. This may help explain why high-fiber diets are consistently linked with lower rates of depression, anxiety, and obesity. In effect, fiber doesn't just feed microbes, it tunes the neural language through which your gut speaks to your brain. In contrast, diets high in saturated fats and processed foods tend to have the opposite effect. These diets promote a less diverse, more inflammatory microbiome dominated by lipopolysaccharide (LPS), producing bacteria which release molecules that compromise the intestinal lining and trigger systemic immune responses. When the gut barrier becomes permeable, often described as leaky gut, pro-inflammatory signals can overwhelm the normal neuropod vagal circuits, distorting the gut's feedback to the brain. This may contribute to the blunted satiety responses and heightened food cravings often observed in diets rich in ultra-processed foods. Additionally, high-fat, low-fiber diets can reduce SCFA production, diminishing the metabolic signals that normally activate neuropods and regulate eating behavior. Neuropod cells themselves are metabolically sensitive. They require a local environment rich in oxygen and nutrients, and they are shaped by the chemical milieu created by gut microbes. Certain bacterial metabolites, such as indoles from tryptophan or secondary bile acids, can either enhance or suppress neuropod activity depending on their concentration and context. This means that every meal shifts the neurochemical landscape of the gut, altering how neuropods perceive the gut environment and how they translate that perception into signals the brain can act on. Probiotics and fermented foods offer another way to influence this circuitry. Live microorganisms like lactobacillus and bifidobacterium are known to interact directly with gut lining cells and modulate the secretion of neurotransmitters. Some strains have been shown to stimulate vagus nerve activity, increase the expression of gut-brain communication receptors, and even change patterns of brain activation in human imaging studies. When consumed regularly, these bacteria may help restore balanced microbial populations, especially after antibiotic use or chronic exposure to a Western diet. More importantly, they may resensitize neuropod cells to beneficial signals, improving the brain's ability to register satiety, safety, and nutritional sufficiency. There is also growing interest in how polyphenol-rich foods, such as berries, olive oil, dark chocolate, and green tea, influence neuropod brain signaling. These compounds aren't fully absorbed in the small intestine and often reach the colon intact, where they're metabolized by gut microbes into bioactive phenolics that can reduce inflammation, support the growth of beneficial bacteria, and protect nerve-like cells from oxidative stress. Some of these compounds appear to directly influence gut epithelial gene expression, suggesting that diet can modify not only the microbiota, but also the gut's sensory machinery itself. Perhaps most fascinating is the plasticity of this system. The gut-brain axis is not static. It's constantly being reshaped by what we eat, when we eat, and how often we vary our food sources. Short-term dietary changes can shift microbial populations within 24 to 72 hours, and corresponding changes in metabolite profiles can alter neuropod signaling almost immediately. Long-term eating patterns, however, are what determine whether the system becomes resilient and adaptive or hypersensitive and dysfunctional. In summary, the diet microbiota neuropod brain pathway represents one of the most powerful and direct routes by which food influences not just body weight, but brain function and emotional state. Through fiber-rich, minimally processed, microbiota-supportive diets, we can foster a gut environment that enables clear, balanced, and timely communication with the brain. This goes far beyond the outdated model of calories in, calories out. It's a vision of food as an instrument of neural tuning, a tool for reshaping how we think, feel, and make decisions about the very behaviors that affect our health.